Hello Year 10, this is a triple science content video, hence the background is pink. So if you are combined science, double science, you don't need to watch this video. If you are triple science, then this is a lesson for you. And this is our final lesson before the Easter holiday, okay? So first of all, to get us thinking about the subject today, we're asking this question: what methods of storing food for long periods of time are used both from times of sailing on ships to modern times? Okay, have a little think about that. Think about what we do, what's the food like in the supermarket to get long life food, and let's get some ideas. So here are our ideas. We can freeze the food. Bacteria can't survive as well. Rate of reproduction is drastically reduced by freezing or even putting in the fridge. We've got cooked food, keeps longer than fresh food. Vacuum packing, we see that in the meat section of the supermarket where the food is sealed tight to remove the oxygen. Tinned food, and covering the food in salt. Okay, so that's just got us thinking about today's topic. We're looking at Decay today. Here's the title, so get those books open, get the date written and the title: What are the best conditions for decay by decomposers? Now it's a bit of an action-packed lesson today. We've got lots to talk about in terms of decay, in terms of anaerobic digesters, which we'll talk about a bit in a while, and the impact of environmental change. So there's quite a lot happening in this particular lesson. I think I've kind of compact more into this lesson because then the next lesson we do is an everybody lesson. It's not a triple lesson, okay? So it's going to feel like it's taking quite a while, but it is quite a crucial lesson, particularly how this anaerobic digester works. It's a really popular subject for the exam board. So really important that we stay focused, get all of our notes done today for this lesson. So what are the best conditions for decay by decomposers? Now we start off by talking about these things here called detritus feeders and decomposers. We tend to think that most of our decay, decomposition is by bacteria and fungi, but we've also got the maggots and the worms. If we think about a compost heap, they are feeding on that matter and breaking it down. Key one for you to be aware of is the bacteria, okay? And if we maybe write some notes here just on that top half of the slide about the decomposers, the bacteria, also the maggots. They're not as keen in the exam questions. If you talk about worms, they're looking for you to talk about bacteria, fungi, maybe maggots. That's the key point. If you need extra time, pause now. Okay, what do microorganisms need to survive? We've got a couple of clues here. They undergo the process of respiration and they need optimum conditions. So what could those conditions, 3 conditions that the microorganisms need to survive? Have a little think, maybe try and list 3 things that could be really important. Think about the kind of work we've done in the past in B4, for example. What conditions do microorganisms, those bacteria, the fungi, need to survive? Let's have a look. We've got oxygen, okay, so that they can undergo aerobic respiration, moisture, some kind of water source, and an ideal temperature, okay? So that links to our optimum conditions. Okay, let's get some notes there then, that the 3 conditions that microorganisms need to survive are oxygen, moisture, and an appropriate temperature. Now, questions that they seem to like in the exam relate to compost storage containers, and they tend to compare a storage compost container that is static, it stays still, to one that can be rotated and the contents tumbled around, and it might have the better one that tumbles, that allows aeration. It might have some holes in it to allow moisture and oxygen in. It might have some kind of more absorbent material, maybe a darker colour material, so that it absorbs more of the heat from the sun and therefore gets the optimum conditions. Okay. So decay in recycling, use of decomposers, not always just about compost heaps, although decomposing plant material, breaking it down and using it as a fertiliser in the form of compost is a great idea. Let's get some notes from this page. But also in sewage treatment plants, okay? And I think you look at this more in, well, I know you look at this more in chemistry, in one of the topics at the end. I think it's C10, could be C9, I think it's C10. Breaking down, using bacteria to break down the faeces, making it safer to release into the river. So it's part of the process to help treat the sewage at the sewage treatment plant, supplying them with large quantities of oxygen for that respiration, so that they can feed off of the faeces, breaking it down and making it safer to release into the river. So let's get some notes from this page. If you need extra time, then pause now. And we've got ourselves an exam question. Gardeners often collect fallen leaves in autumn and place them on compost heaps. So there's the leaves fall from the tree, the gardener collects the fallen leaves, the leaves are placed on a compost heap. Over the next year, the leaves decay. Which living things cause the decay? Okay, let's get an answer in our book. Which living things cause decay? We covered that already. The leaves decay more quickly in summer than winter. Give one reason why. Why are they going to decay more quickly in the summer than in the winter? Give one reason why. We've also talked about that so far in this lesson. The compost heap has holes in its sides to let gases enter. Which gas is needed for decay? And we've covered that one already in this lesson, so this should be quick recall of things that we have already covered. Okay, let's go for our green pen then. So the living things which cause decay are the microorganisms, the microbes, the bacteria, the fungi, you might have called them pathogens, the decomposers, or an example such as mould. We've got, see here they're not keen on the word worms, so that's what I was saying before to be careful about. For B, why does it decay faster in the summer than the winter? Because the weather is warmer, hotter. They like these kind of comparative words rather than just saying it's hot. If you say it's hotter, you're comparing it to winter. Okay, so that's why the ERs are there. In the winter it's colder. You cannot write that the heat dries the leaves out. Okay, it's to do with creating the optimum conditions for those enzymes. Okay, we link a lot back to enzymes. If it gets too hot, then obviously we've got the denaturing of enzymes. And the third question was which gas is needed for decay, and that is oxygen. So let's do some green penning there. And if you need more time, pause now. Okay, another exam question. Gardeners often put waste materials onto compost heaps. The graph shows how the conditions in a compost heap affect how quickly waste material in the heap decay. So we've got rate of decay in arbitrary units. Now arbitrary units is, it means there is no unit, okay? It's kind of like a scale. Like if I said, how happy are you on a scale of 1 to 10? That's arbitrary units. There are no units, it's just kind of relative to each other. So we've got two temperatures there, 15 degrees and 25 degrees, and then at the bottom of the graph we've got percentage concentration of oxygen. Describe the effect of increasing temperature from 15 to 25 degrees C on the rate of decay at 20% oxygen concentration. Okay, so we've got 20% oxygen concentration here, 15 degrees C, 25 degrees C. Okay, if you can just try and process where that is. Okay, 2 marks this one is. And let's move on. Gardeners are advised to put waste materials into special compost bins. These bins have holes in their sides. Holes in the sides of the compost bin help the waste materials to decay faster. Explain why. Again, we're writing this answer down. Quick 2 marker. And part B. A gardener noticed that some of his plants were growing poorly. He put some decayed compost onto the soil around the plants. 6 months later, the plants were growing well. Explain why. Okay, so 1 marker. And if you need any extra time, pause now. Right, let's go to our green pen then. So green pen's out again. So there is a increase or higher or faster, quicker. So 1 mark for saying that at the higher temperature of 25 degrees C, there is a faster rate. And then if you use some data from the graph, it's twice as much, or it doubles. It goes from 30 to 60. 30 at 15 degrees C and 60 at 25 degrees C. And then we needed why there were the gaps in the container there to allow oxygen in or to allow air in, and for the microorganisms, bacteria, microbes, fungi, decomposers, whatever you want to refer to them as, to use it for aerobic respiration. Okay. So 2 from that list. You might have got one of the others, but I think I've chosen the most popular ones. And then part B, compost contains the minerals, the nutrients, the elements, the ions. You might have said nitrates or named a particular mineral, sorry about that. Not glucose, okay? Glucose is made during photosynthesis. It's not something that comes through the soil, and people tend to forget that, okay? So the glucose comes from photosynthesis, not through the ground, okay? If you need extra time to green pen, pause now. Okay, we're looking at this stuff called biogas now, okay? And you may well have come across biogas. We've been talking in chemistry about bioethanol maybe, which is, and we talk about it again in this topic anyway, it's where we take sugar cane and we ferment it and we get bioethanol from it, which can be used as a fuel. We've also got biodiesel, and you see there is trucks on the road that say that McDonald's actually filter all of their cooking oil and then they use it in their trucks as a fuel, and it's called biodiesel, okay? Now we're looking at the production of biogas, okay, which is very high in methane, some carbon dioxide, which isn't useful, it's the methane that's useful, and then there's other trace gases as well. And this is an anaerobic process, so it's in the absence of oxygen, and it takes place by bacteria. And you may have seen near the Hatch Warren roundabout, it says that there's a brown sign with a truck on it, and it says anaerobic digester that way. That is what we're talking about. We're talking about anaerobic digesters. So this kind of machinery, if you like, is huge, okay? It's called a continuous flow process. Now what you might want to do is just note down what the time is at the moment on the video, because I'm going to talk through it, and then you might want to go back through it again to get your notes written down. So if we just kind of concentrate first of all, get the idea, get the principle, and then you might want to go back and watch again to get your notes. So this is a continuous flow process. That means that substances are continually being fed into the digester and substances are continually being removed from the digester. and the main important product that we get from it, although we get two useful products, is biogas. Now this container is under the ground. It's kind of made of giant concrete structure. It may well in colder climates be heated so that we're getting those right conditions, the optimum conditions again. And into this part here is the material, any organic material. Organic material means grass cuttings, plants, anything that was living and can decompose. It could be vegetable peelings. You know when the bin men come and they collect the your green garden waste in the green sack. This stuff is going to go into this anaerobic digester. Okay, so it's constantly being fed in. This thing here kind of floats up. So as the biogas is created in this area by the bacteria, this will float up and it will, you'll see that there's gas in there, and when it rises a lot, there's a tap here. You can actually tap off the gas and collect it here. And over here, this is the waste product coming from the digester, and this is called slurry, and this is actually used as compost. Okay, so they'll bag that up, the slurry, and use it as compost. Okay, so let's talk through that process now. So there's our organic material going in. Maybe a good idea, so you can take like half a page in your book, draw a picture of the digester, and then you can annotate around it with all the key points. So we've got the organic waste going in. That's anything that can kind of be broken down. Here they've suggested it's domestic waste. We talked about that. That's the grass cuttings and stuff. Also agricultural waste. So waste from sugar factories or even sewage can go into here as well. Okay. And then we've got this bacteria respire anaerobically in the absence of oxygen. That's why it's quite useful that it's a continuous process, because if it was a batch process where you kind of start it up, let it run and then stop it, every time you start it up again, you've got oxygen in there that's you've got to wait for that to be used up by the bacteria to then get it back into anaerobic conditions. So when they initially set this up, there will be oxygen present, therefore the bacteria will be respiring aerobically and carbon dioxide will be produced. Well, they don't want carbon dioxide, they want biogas, they want this high methane. So by keeping it in anaerobic or waiting until it gets into these anaerobic conditions and then leaving it there by continuously feeding it and taking away the biogas and the slurry, we're able to keep it in better anaerobic conditions in the absence of oxygen. So bacteria respire anaerobically, produces the gas, which lifts that floating gas holder, okay, just here, up. And we've got that tap there, so that we can kind of tap it off really. And then the biogas can be extracted and used when required, mostly for producing electricity. Okay, and although that biogas will be burnt, because it originally came from plant form, which was went through the process of photosynthesis, it's carbon neutral. So it's releasing carbon when it's burnt, but as a plant it took in carbon in the form of carbon dioxide, and therefore it's a carbon neutral process. Again, this also comes up in chemistry as well, but we just talk about it in this topic too. So we've then got the slurry, used as a fertiliser, very high in nutrients, okay, and that's tapped off on the side there. Temperature is important. We need to keep those optimum conditions. Obviously those bacteria will release some heat, so long as it's well insulated, that might be enough to maintain the temperature. Colder climates, then that could well be heated. And then biogas is produced on large scale. I talked about it being a continuous flow method. We're actually going to look at another process, how a substance called mycoprotein is made right at the end of this topic, and it's a batch process that's aerobic. So we've got this process that's anaerobic and continuous, and we're going to kind of compare that to another method that is aerobic and batch later on in the topic. So they're constantly adding the organic matter, removing that slurry, removing the biogas, okay? So that is how an anaerobic digester works, okay? Now at this point, you might want to go back, I think it was like minute 14, something like that, and you might want to go back and just kind of improve your notes. This is quite a good one to be on at the end to get that summary, okay? If you're not going to move on, if you're not going to go back, that's fine. If you are, then do that now. I'm just going to, give me a sec, I'm just going to try and, I've lost the mouse, it keeps coming back and forwards. Okay, this is just me, I took some screenshots to explain it in case the video didn't work. Okay, last 3 slides I think. So the last bit just kind of tags on the end. It's not really that related, but you can't make a whole lesson out of it, so it's just a couple of key points here. Impact and change in temperature. So we know that global temperatures are changing, and one of those changes in temperature could be seasonal changes, and we've talked before in this topic actually about bird migration patterns, and as temperatures are changing, the migration patterns are changing. So they seasonally migrate to warmer climates, but actually now that we're seeing some changes in when that migration takes place, okay? May be useful to get some notes from this slide here. And if you need extra time, pause now. Okay, impact in the change of availability of water. We've got the seasonal changes, animals and plant species change between wet and dry seasons, so you might see different plants or no plants. Distribution of animals, if there's no water then they're going to keep moving, so wildebeest migration changes as rainfall patterns change. Okay, that's just an example, and we'll take some notes from this page. And then if you need extra time, pause now. And the last slide, okay? These are lichens, and lichens come in 2 forms. There's kind of this crusty, their words, not mine, crusty lichen, which is quite flat. Sometimes on walls it can look a bit like a bit of old chewing gum that's kind of squashed into the wall, but it's actually a lichen. And they do not like poor air conditions, okay? So they like air that is very clean and fresh. So if you see lots of these lichens growing on trees and on walls and maybe, you know, side of the house or something, it means the air quality in your area is really good, okay? Where we see the absence of the lichens is where there's lots of sulfur dioxide. Now, when cars are burning petrol, there are impurities in that petrol in the form of sulfur compounds, and when they burn they form sulfur dioxide. You've probably heard of that, leading to acid rain. And the sulfur dioxide is at really kind of low concentrations. It's hard to monitor how much they're changing, so we use the lichens as an indicator species. That means that, say there is a road built somewhere, we can see over time by using our transit line that we talked about the other day, and on our quadrat, we can see how much the lichen populations have decreased over time at different distances from that road. So it might be that really close to the road the lichens have completely been wiped out. It might be that further away from the road we see that they've been unaffected. We've then got bushy lichens, again their word not mine, and bushy lichens, they like air pollution, okay? So sometimes they can replace the crusty ones in those areas where sulfur dioxide is built up, okay? So maybe have a look, see if you can see any on trees or in buildings or whatever, and you'll know what I'm talking about. But good example of how a transit line can be used. They're used as an indicator species to look at the impact and change of the atmospheric gas sulfur dioxide. So that is the end of our lesson. That is the end of all of our biology lessons now for a couple of weeks while we've got Easter holiday. If it is that you do want to do some more work, there are loads of videos on my YouTube. They're all in playlists for different topics. So if you feel like doing some revision for the end of year exam, which at some point in time we will have that exam, it will be on B1, B2, B3 and B4. If we have that this side of the summer holiday, then that's when it will be. If we don't, we will have it in September, and that's going to be a really important test for us, okay? So you could be looking at any of those B1, B2, B3 and B4 playlists and getting some revision done if you're at a loose end over the half term, or the end of term I should say. Hope you found that useful. Know it was a bit longer than normal, but hopefully it was quite interesting, and now you know what that anaerobic digester is all about. Hit subscribe, hit share. I'd really appreciate that, and then we can let more people know about our distance learning lessons. Okay, thanks very much. Bye.